drug treatments as a way to control it. Right? So the vaccine that's used is called a BCG vaccine. It's actually not TB itself. It's a very close relative to TB called Mycobacterium bovis. Okay? If you have a name like bovis, what do you think it affects? Yeah, cow, livestock. Okay, all right. But when this vaccine based on this will give you an immune response that also works for TB. So typically in high-risk countries, this is where you use the vaccine approach to try to control disease. Okay. Any questions about this? All right. So this is serious pathogen number 1. This is another one that we've talked about elsewhere in this unit. Now, remember, we talked about anthrax as being a skin infection. Here, we're going to look at it as a respiratory infection. Okay? And this is what gets a lot of the media coverage when it was an issue, because this is also the form that's used in terms of, let's say, bioterrorism. Okay? So it would be like a powder that would be spread throughout a building, and that could be inhaled and cause this form of anthrax. So we're looking at inhalation anthrax. All right, so just to go over some things we looked at earlier, Bacillus anthracis, it forms endospores. It also makes a very potent toxin, anthrax toxin, and this is what kills human cells. It just outright kills cells. Inhalation anthrax, this results from inhalation of endospores. And what happens is when an individual inhales endospores, they initially have flu-like symptoms, but then they start to go into serious health issues with severe breathing problems and then go into shock. Okay? So at first it resembles like a bad case of the flu, but then it gets much, much more serious. And typically untreated cases of inhalation anthrax are fatal. Right? So there's actually a high fatality rate associated with untreated cases. It's like 90% plus, right? Which is why this has such an appeal as a bioweapon. Right, so think about it, it's endospores. Endospores can survive lots of adverse conditions, okay? And you can easily spread it. You can spread it as an aerosol, right? So it has a lot of potential worries for those of us on the other side trying to defend against this kind of weapon. The hard part is, though, you have to be able to make the endospores into a form that you can like spray. It actually takes very sophisticated equipment. Right. Any questions about this? So how do we treat, if someone has inhalation anthrax, how do we treat it? What can we use? So what do you typically use for these types of microbial infections? So when you think of bacteria, what do you think of? Antibiotics. Antibiotics. Okay, so there are some antibiotics that and this is in part because of 9/11 and what happened after 9/11, where we started to see those cases of anthrax. Right, that was way back when. All right. That we know with the case of inhalation anthrax that there are antibiotics that can work. All right, any questions about this? All right, let me. So now I want to go into another serious pathogen, another serious respiratory pathogen. This is one that we're always on the alert for. Okay, influenza. So every year you hear about influenza. And hopefully, after this, you'll see why we're so worried about this. Right? Because all of us think about the flu as being, it's just the flu. But there are many examples, numerous examples, of types of flu viruses that are very deadly. And so this is why we actively monitor influenza. Right? In fact, because of the technology now, actually, and also because I see all these screens around. Right? One of the current strategies being used is to use social media as a way to try to monitor flu epidemics or potential flu outbreaks. So if you think you have flu symptoms, or you look around you and you think that the person next to you has flu symptoms, you can actually report that. Right? There's a website you go to and you just say, yeah, so right now, since tonight I can go home and say, so in my class, I think has the flu. Yeah, you can go, you can report that, and if they see a trend, right, because they can monitor where all these reports are coming from, that will alert them to investigate further. Okay, right, so in fact, they're finding that looking at the internet, looking at how people track the web for flu is a far better predictor than using the conventional scientific methods that we use. Right? So we just look at how people use Google, and that seems to predict flu outbreaks better than our current surveillance methods. Yeah, because so if people are getting sick, they say, okay, flu. I'm going to look up flu on Google and see what I can do about my sickness. Right? So when they monitor stuff like that, searches for flu virus, they seem to be able to spot very accurately where the outbreaks are occurring. Okay, all right. So I guess it's just not. I guess Google is not a total waste of time. All right. Okay, so influenza. Getting back to the virus. This is an envelope virus. Okay, I have a picture right here. So here is the envelope. Okay. We're going to be spending some time looking at these little knobs at the end. These are the proteins. These are two of the proteins, two of the important proteins that we find on influenza. Before I get there, let me just go through the list here. The genome has 8 single-stranded RNA segments, so it's a segmented genome. We're going to find that's an important issue, the fact that it's segmented, because what that means is that this allows the potential for this virus to mix and match segments with another virus. Okay? All right, so it's hard to do that if you only have one segment. But if you have these little segments, right, you can start to mix and match your genetic segments. And we'll see what issues that raises. Now, the proteins that I mentioned, there's 2 proteins. And some of you may have heard about the abbreviations for these proteins. One is called hemagglutinin, or H, HA or H. The other is called neuraminidase, NA. So, some of you remember this virus. It's from influenza, the one that seemed to cause all the worries a few years ago, H1N1. Right. So yeah. So it's related to swine virus, although it actually has elements of all types of virus. It has a little bit of bird, a little bit of human, a little bit of swine. Okay. All right. So H in this case refers to what? Hemagglutinin, right? And refers to neuraminidase. That's why we have these. That's why we have these letters. Okay. It refers to these 2 proteins, and it refers to the type of protein. One, right? H, 1 for N. Okay. All right, so it turns out that there are various forms of hemagglutinin, various forms of neuraminidase that are found on the virus. Okay. Right, and so we can use that as a way to differentiate one form of influenza from another. What these proteins do, just to give you a function for this. Hemagglutinin, this is what binds to epithelial cells. Okay, so this is what attaches to your cells, right? Like your lung cells, let's say. Neuraminidase is what hydrolyzes the mucus in lungs. Okay, so it helps. Neuraminidase helps the virus be able to spread. Now, beyond H and N, there are 2 types of viruses that are responsible for a lot of the human epidemics. That's A and B. A is usually the one that's more predominant. This is the one that we're usually more scared of, is A. When you get a flu vaccine, right? So when you get a seasonal flu vaccine, okay, so let's go through this. When you get a flu vaccine, how many viruses do they actually give you? Do they actually inject you with? Yeah, that's several. It's more than one. We don't know how many. Yeah, so they give you 3. Okay? They give you 2 type A, and then 1 type B. Okay? Right, those are the viruses that they use. Okay? Right? So so H1N1, what kind of virus would that be, A or B? Based on what we know so far, try to describe them. It's A, yeah. So one of them is actually H1N1. I can't remember what the other one is, and then they have one type B. Okay. All right. Any questions about this?
So is the A and B like shape, or how do they differ?
Yeah, so this refers to, A refers to hemoglobin and B refers to neuraminidase. So what happens is that you can have slight mutations in the structure, slight changes in the structure, right? And so how would you tell